Welcome to my channel Sehba Medical Academy. So today we will start a new topic, gram positive versus gram negative bacteria. You know, in the invisible world of bacteria there is a huge divide, and it will come down to one very small structure difference, but there is a small important part that one different can divide if an infection is mirror or life threatening. So today we will going to understand this divide. This is a question that affect all of us, right? Why it is that one bacterial infection can be treated with a simple pill, but another one fights back against everything we use? Well, the answer is not about what bacteria do, it's about how they are built. Yes, the real secret is not their tricks, it is written in their structure. I am talking about their cell walls, the protection they are used against everything. And remember, we are in a constant battle with them. We make a new drug and they develop new defense. This is the problem of antibiotic resistance, and the winner of battle often depends on their structure. To understand this, we need to go back to the 1880s. Our story starts with a Danish scientist, Hans Christian Gram. He was not trying to change medicine, he was just solving a simple lab problem. In 1884, he was looking at lung tissue from pneumonia patient, but the bacteria were almost invisible. They were mixing the human cells. He just wanted way to make bacteria clearly visible, so he created a simple four-step method. First, he added a purple dye called crystal violet. Then he added iodine to lock it in. The most important step was washing with alcohol. Finally, he added a pink dye. Logically, all bacteria should look the same color, right? But that is not what happened. Some bacteria stayed purple while others lost the purple and became pink. At that moment, he had unknowingly discovered a major difference in bacteria. The big question was why? It took scientists many years to understand this, and the answer is in the cell wall. It is the protective layer around every bacterium. To understand this, we need to look closely at its structure. So, everything depends on one material, and peptidoglycan. Think of it like a chain link fence. It is a strong mesh made of sugar and proteins. It gives bacteria their shape and prevents them from bursting. And the amount of this material is very important. This helps us understand gram positive and gram negative bacteria. There are many differences, but two are most important: the thickness of peptidoglycan and whether there is an extra outer membrane. First, peptidoglycan, gram positive bacteria have a very thick layer. It is like strong armor, but gram negative bacteria have a thin layer. This is a big difference, but the next feature is even more important. Before that, let's quickly understand the cell membrane. It is a phospholipid bilayer, a very smart design. These molecules have head that like water and tail that do not like water, so they form a flexible barrier. Now remember this, because gram negative bacteria have an extra outer membrane on top. Think of it like this: one building has a single wall, but another has an outer wall plus inner protection. This is much stronger defense system, and this explain grams result. In gram positive bacteria, the purple dye goes deep into the thick wall. It's get trapped there. Iodine looks in it in place, so when alcohol is used, the dye does not come out. The cell stays purple. In gram negative bacteria, the outer membrane block the dye. Only a little enter and it is easily removed by alcohol. So the cell become colorless. Then it absorbs the pink dye, so it appears pink, mystery solved. This is not just a lab concept, it is a very important in real life. This structure difference explains why some infections are harder to treat. Now we are back to the antibiotic battlefield. That outer membrane is a strong field. Outer membrane is a strong shield. Many antibiotics cannot pass through it. Their defense system is very powerful. First, the membrane itself is a barrier. Second, it has toxic molecule that can cause serious condition like septic shock. And third, even if antibiotics enter, they have a pump that push them out. This make them extremely effective. So think about it, a simple lab experiment lead a major discovery. Today, with rising superbugs, we are facing a serious challenges. Gram-negative bacteria have a very strong defense system. They not only block antibiotic with their outer membrane, but they also have a special protein channel that control what enters the cell, an enzyme that can actually break down antibiotics. Even if a drug managed to get inside, their pump can push it right back out. So the big question is, can we design drugs that are smart enough to break this gram negative defense? Thank you for watching. Please like, share, and subscribe to my YouTube channel, Sehba Medical Academy.